membrane is done. Function of cell membrane is done. And associated with cell membrane what? The solutions, permeability we have studied. No? The transport functions of cell membrane. What were the transport functions we had studied? Cell membrane is semi-permeable, so it will allow the movement of only some particles, movement of water. Cell membrane is semi-permeable, it will allow majorly the movement of the water molecules. And the movement of the water molecules under different conditions. So we studied if the cell is in a dilute solution, hydrochloric solution, then what will happen? If the cell is in a dilute solution, what will happen to the cell? The cell will swell up. And take out your notebook and check what was the last topic we completed. Okay, so we will start with so the concept of the cell membrane we had studied already, the functions of the cell membrane we had studied already. Let us start with the other cell organelles. The functions of the cell membrane are done. What is the function of the cell membrane? I will just now revise it. The functions of the cell membrane is cell membrane helps in transport. Cell membrane is semi-permeable, so it allows the movement of the water molecules, and based on what type of an environment the cell has. If the cell has a very dilute environment around, then the water molecule will enter the cell, making the cell swell up. So the solution surrounding at that time will be termed as hypotonic. If the cell is shrinking, meaning the cell is kept in a hypertonic solution, meaning the cell is losing water. Hypertonic solution we have to find. We understand that the cell is in a solution or any particular plant part in the soil, in the water, but then the cells are not showing a major change. There is no change in the shape of a cell. And what type of a solution we have to understand surrounding the cell? Isotonic. Meaning the cell is neither gaining anything nor losing anything. So that was what about the basic concept of permeability. Let us talk about the other cell organelles today. Did you complete the diagram check? Yes. Or else, as we draw the parts, we have to finish up the diagram. So let us talk about the nucleus. We talk about the nucleus and the other cell organelles. We start with the nucleus. This is the nuclear membrane. Check if you have marked in your diagram. Yes. The nuclear membrane. These are the chromatin threads. We talk about the nucleus. Let us take the next cell organism, that is the nucleus. What are we going to study? For the nucleus, we have to study the structure, the function. So for the structure we talk about, the nucleus, what is the basic structure of the nucleus? The structure of the nucleus is present both in the plant and the animal cells. Nucleus will always have a double-walled nuclear membrane. The nuclear membrane is always double-walled. But the nuclear membrane is porous, meaning if you see the nuclear membrane, it is not a continuous structure. It has got gaps in between, and the gaps are actually meant for movement of substances inside and outside the nucleus. So when we are looking into the structure of the nucleus, what are some of the features? It has got a double-walled nuclear membrane. The features are, it has a double-walled nuclear membrane. You will write down all these points that I am giving you. Double-walled nuclear membrane. Then, the nuclear membrane is porous. The nuclear membrane is porous. Meaning what? It has got pores which allow the movement of substances. Nuclear membrane is porous. Third is, the inside of the nucleus, there is a matrix-like substance. What is the matrix-like substance known as? Nucleoplasm. Nucleoplasm is the matrix inside the nucleus. Nucleoplasm is the matrix inside the nucleus. And what is suspended on it? Chromatin fibers are suspended. Chromatin fibers are present. The chromatin fibers are present inside the nucleus. So when we are talking about the chromatin fibers, what are chromatin fibers and what is their job? So talking about the chromatin fibers now. These chromatin fibers are in the form of some fine, long, extensive thread-like structures. But these are extensively long. But this same chromatin thread, when they are extensively condensed, we come across that this entire structure, which is now appearing shorter and broader because it is extensively coiled, then what is this particular structure known as? This particular structure would be known as the chromosomes. These are known as chromosomes. So the upper diagram and the lower diagram are same. Composition is same, but only that this is extensively long uncoiled, and this is extensively coiled and compact. Composition is the same. This is known as chromatin fiber. This is known as chromosome. So now, when we are talking about that, this chromatin fibers of the chromosome, where are they present? They are present in the nucleus. So I would say that these chromatin fibers, next is what? The chromatin fibers condense or coil. The chromatin fibers undergo the process of coiling. The chromatin fibers undergo the process of coiling to form chromosomes. They undergo extensive coiling, and then when they undergo extensive coiling, what are they known as? These are known as chromosomes. So when we are talking about chromosomes and the chromatin, is it the same thing? Yes, it is the same thing. Only that I talk about a young cell and I talk about a mature cell. Something like, if I talk about a young cell and in that young cell we come across the threads are like this. We don't understand what is which one, how many are there. But the same cell, when it is now mature, ready for the process of division, and we get to see that here this is the nuclear region, you can generally count them also. You can generally count them also because they are extensively coiled. They become separate units and you can see them under the microscope. You can see them. What are these chromosomes made up of? Now, every single chromosome that we understand, every single chromosome is made up of a helical double-stranded structure. A helical double-threaded or double-stranded structure. And what is this helical double-stranded structure known as? this helical double-stranded structure is known as the DNA. So this helical double-stranded structure is known as the DNA. So when we are talking about this chromosome and the DNA, the chromosome and the DNA would be something like this, that the DNA structure inside the chromosome. So we understand that what is the DNA and the structure of the chromosome. Or do plant and animal cells both have this? Yes, both have this. When we are talking about the structure of a cell, whenever look into the structure of a cell, any cell we talk about, any cell will generally have the chromosomes. And try to look into what is present in the chromosomes. I want to magnify this structure and see they would always have a ribbon-like structure like this, a ribbon-like structure like this. So I have written down, see, they double helical, they are helical. What is meaning of helical? Wavy there. And they are double-stranded too, made up of two strands. Double-stranded. So they are double-stranded helical structures, and this double-stranded helical structures is what is known as the DNA molecule. These are all the DNA structures that we talk about. These are all the DNA structures that we talk about. So this is the DNA. What is the full form of the DNA? Let us try understanding the full form of the DNA. What is DNA and what is the basic term? So this entire structure, if I now talk about that, how does the DNA look like? The DNA is always helical like this. The DNA is always in the form of a ribbon-like structure. And this DNA, which is in the form of a ribbon-like structure, this always has a specific composition, of course, is there. Now, we are not going to talk about the composition right now. So here I am saying that what is a chromosome? What is the definition of a chromosome? Chromosome is a extensively coiled chromatin fibers. Chromosomes are extensively coiled chromatin fibers. So these chromosomes that we are talking about, chromosomes consist of extensively coiled chromatin fibers. And what is the composition of the chromosome? Composition of the chromosome is a complex molecule which is helical, which is made up of two strands. It's a ribbon-like structure. What is that known as? DNA. What is the full form of DNA? Deoxyribonucleic acid. What is the full form of DNA? You'll have to note this. What is the full form of DNA? Deoxyribonucleic acid. It is a complex molecule and it is acidic in nature. So look into the structure of the DNA now. The DNA is always like this, helical and flat and ribbon-like. But this entire DNA, when I see, it has got certain units on either side, and then two thread-like structures, and then there are some units which are bonded like this, okay, with the help of some bonds. So now, I look into each particular segment, say suppose this, and say suppose this. It's a small segment of the DNA that I'm talking about, a small segment of the DNA. So this segment of the DNA, I may understand, this is responsible for our height. Maybe this segment of the DNA is responsible for our eye color. Yes. Now this particular segment of the DNA may be responsible for shape of the nose, etc., etc. There are so many characteristics to talk about. So what am I saying? I'm saying that this entire DNA that we are talking about, the ribbon-like structure, the molecule that I'm talking about, where is it present? Inside every chromosome. And when you see the detailed structure of the DNA, it is understood that every small region of the DNA is regulating one particular characteristic that we show. So every small segment of the DNA, every small segment of the DNA is responsible for every specific characteristic that we show. So now, what is that small specific region of the chromosome known as, or that small specific region of the DNA known as, that is known as the genes. What is the meaning of the word genes? What are you supposed to understand? The meaning of the word genes is Meaning of the word gene is what? It is a small segment of the DNA which can regulate our different characteristics. It is the specific segment of our DNA. So, see here. So when we are talking about what is meant by the word gene, it is a small segment of the DNA. And like this, there are hundreds of segments all over the DNA. Every single chromosome I speak, every single chromosome will have this type of ribbon-like structures. And in that ribbon-like structure, there will be small segments in hundreds. And every single segment is responsible for one particular character. Now, one of those characters I'm thinking of, the texture of the hair, the color of the eye, the color of the skin, or internal factors also, something like the release of certain enzymes, release of certain pigments. And also the third type of characteristic that this gene regulates is our behavior. Each one of us has got different types of behavior. I talk about the tolerance and the patience and the, when I talk about human behavior, that is also because of these genes that we have. And this is what we inherit from our parents during the process of our formation. So when we are talking about this, what is this? This is a DNA molecule. But where is this DNA molecule present? In the chromosome, in the DNA. Where is the DNA present? In the genes. Then how do we define what is a gene? Gene is a segment of the DNA that regulates our character. What is this entire ribbon-like structure known as? This is known as the DNA molecule. What is its full form? Where is it present? Inside the chromosome. So now if I ask you, what is the chemical composition of the chromosome and the chromatin fiber? What is the chemical composition? Chromosome and the chromatin fiber? It is the same thing, DNA. So what is DNA? What is its chemical formula? The full form, right now? What are chromosomes composed of? Right, one question. What are chromosomes composed of? What are chromosomes composed of? In this question, what are chromosomes composed of? What are chromosomes composed of? Write its full form. What are chromosomes composed of? Write its full form. Full stop. What should be the answer of this? What is the first question I gave you? What are chromosomes made up of? DNA. Chromosomes are made up of DNA. Write down and then write down the full form. Chromosomes are made up of DNA and then write down the full form. Okay. Next definition. Define a gene. Define a gene. Define a gene. Right now, a gene is a specific segment of. A gene is a specific segment of the DNA molecule. A gene is a specific segment of the DNA molecule segment of the DNA molecule, which encodes for a protein, encodes means carry information for a protein, which encodes for a protein and regulates specific characteristics, which encodes for a protein and regulates specific characteristics So we are talking about the nucleus, where you are supposed to know what is the basic structure of the nucleus. What are the terms? What are the parts you are supposed to know? The nucleus consists of a double-walled nuclear membrane. So if it is the membrane, then what must be the composition of this membrane? Same like this. Lipids and proteins. So what is the composition of the cell membrane? Ever asked? You understand what is the composition of the cell membrane to write lipids and proteins. What is the composition of the nuclear membrane? Same, lipids and proteins. And this nuclear membrane is porous. I mentioned that nuclear membrane is porous. What is the meaning of this? Nuclear membrane porous, meaning it has got gaps in between. It is not a continuous layer, and these gaps are meant for transport of substances in and out into the cytoplasm. What else is there in the nucleus? It has got the matrix. What is the matrix of the nucleus known as? Nucleoplasm. What is nucleoplasm? Right on the next question. What is nucleoplasm? What is nucleoplasm? What is nucleoplasm? Check the spelling. What is nucleoplasm? The gel-like substance inside the nucleus. Gel-like substance inside the nucleus, which consists of gel-like substance inside the nucleus, which consists of gel-like substance inside the nucleus, which consists of proteins, salts, and various other substances dissolved, and salts, SALTS, salts and various other substances dissolved is known as the nucleoplasm. Next, I have been talking about the chromatid and the chromosome. So, other features of the nucleus, what to write? What is the structure? It contains the chromatid fibers. It contains the chromosome. But in your exam, you will also get a differentiate between chromatid fiber and chromosome. Then what I need to write? Chromosome contains chromatid loose and long thread-like structures. Thin. Chromatid not visible under the microscope. Chromosome distinct under the microscope. Chromatid present in a newly formed cell, young cell, and chromosome present in a mature cell. Okay, right on the next. Differentiate between, make a table, differentiate between chromatid chromosome. Differentiate between chromatid chromosome. Make a table and write down. chromatin and chromosome, right? Chromatin and the chromatin. Under chromatin, extensively long, thin, thread-like structures. Extensively long, thin, thread-like structures. Present in the nucleus. Extensively long, thin, thread-like structures present in the nucleus. Chromosome. Or chromosome. Chromosome. Under chromosome, right? They are highly coiled. Chromatin fibers. They are highly coiled chromatin fibers. They are highly coiled chromatin fibers. Point number 2, for chromatin. They are, why so late?
You can know the timings.
Your lecture starts from 2:45. We are not gonna late. Check your timetable. Okay. Now chromatin, second point. Let's say they are present in a newly formed cell. They are present in a newly formed cell. They are present in a newly formed cell. You write down the points I am giving them. I have already given them some 2-3 questions that you can take down later. Now you start with the table difference between chromatin and chromosome. The first point is over, so write from the second point. They are present in a young newly formed cell. The other side, they are present in a mature cell, chromosome. They are present in a mature cell. They are present in a mature cell. Is this the bio notebook? No. Now you write here today, tomorrow you write somewhere. When I ask you where we are done, you're not able to tell me. Maintain a proper notebook. Maybe you have the previous notes you have joined right from the beginning. You have the previous notes? Yes. So that notebook, all these things should go there, and next class that same thing. They are present in a mature cell ready to divide. They are present in a mature cell ready to divide. Number 3, under chromatin. Under chromatin, they cannot be, or they are not visible. They are not visible under the microscope. Not visible under the microscope. Not visible under the microscope. Other side, chromosome, clearly visible under the microscope. Clearly visible under the microscope. Clearly visible under the microscope. So now, the difference between chromatin and chromosome should be clear. Chromatin are fine long, chromosomes are condensed. The last one more thing that you have to remember is what are the carriers of heredity? Meaning what we are talking about, that these chromosomes or the chromatin, these are known as the hereditary units. What is the meaning of hereditary units? These chromosomes can move from one cell of the parent to the cell of the offspring. How? Because whenever I talk about, say suppose I talk about 2 cells, and now that I understand the cell of one parent, the cell of the father, I am talking about the reproductive cell of the father, the reproductive cell of the mother. And this says the reproductive cell of the mother and the father during the course of fertilization results in the formation of the zygote. And so that is how, now this is the cell of the zygote, and this zygote will ultimately result in the formation of a offspring. What is the meaning of the word offspring? The next generation organism. From here, the next generation organism will be formed. So here, this is the concept of heredity. What is the meaning of heredity? The chromosomes are moving from one generation to the other. The chromosomes are moving from one generation to the other. The chromosomes are carrying the genes, and that's the reason the characteristics pass on from one generation to the other. So if you talk about yourself, if I think of myself, yes, we bear some resemblances with our parents. How? Because the characteristics are passing on from one generation to other by the way of genes. So what are genes we have defined just now? These are units of the DNA molecule which regulate our characteristics. So what are hereditary units? If you get this question in your paper, what are hereditary units? Hereditary units are what? Hereditary units are the chromosomes. Why are the chromosomes known as the hereditary units? Because they carry genes. What are the chromosomes made up of? DNA, and DNA carry genes. So what are hereditary units? Name the hereditary units in living organisms. What are the hereditary units? Chromosomes. What are their functions? Why are they named as such? Because they carry genes from one generation to the other. And that's the reason whenever any particular generation compare characteristics with the previous generation, definitely there's a resemblance. Okay? The last question for nucleus we will take. What are hereditary units? Right now? Or you write, why are chromosomes known as hereditary units? I think that would be better. Why are chromosomes known as hereditary units? Why are chromosomes known as hereditary units? Better. Chromosomes are known as hereditary units as they pass on. Chromosomes are known as hereditary units as they pass on from one generation to the other, as they pass on from one generation to the other. That is from the parents to the offspring. From the parents to the offspring. From the parents to the offspring. Full stop. So when we are talking about why are chromosomes known as hereditary units? They pass on from one generation to the other, from the parents to the offspring. Full stop. Chromosomes carry genes. Continue. Chromosomes carry genes which are responsible for. Chromosomes carry genes which are responsible for chromosomes carry genes which are responsible for regulating different characteristics in the offspring. Different characteristics in the offspring. Okay, so now to talk about the functions of the nucleus also. We have to talk about the function of the nucleus. What are the functions of the nucleus? We have to talk about the functions of the nucleus. The major functions of the nucleus also known as the director of the cell. What is the meaning of the word director? Meaning it has the capacity to regulate all the functions of the cell. This nucleus, we had learnt about the chromatin, we had learnt about the chromosome, we are talking about the genes, we are talking about the DNA. Apart from all these regulating the characteristics of an individual, inside the cell, what are the other functions it is doing? This nucleus can regulate the functions of all the other organelles. So when we have to mention what are some prominent functions of the nucleus, number one, nucleus can regulate all the general functions of the cell, meaning it can regulate the endoplasmic reticulum also, the mitochondria also, the lysosome also, the Golgi also. Every particular cell organelle is actually dependent on the nucleus to carry out its functions. This is one. Number two is what? Just what we have written about the chromosome right now. That this nucleus can regulate the passing over of the characteristics from one generation to the other. How? Just like you know about the genes and the chromosomes. So nucleus can regulate the passing over of the characteristics, transference of the characteristics from one generation to the other. Number three, it is the nucleus which can also regulate the concept of heredity. So when we are talking about the basic functions of the nucleus, what is it doing? Inside the cell, it regulates all the functions. And if I talk about an organism, it is the nucleus which can regulate the transference of characteristics from one cell to the other, carries out the process of cell division. So prominent functions of the nucleus, three major functions of the nucleus, let us now write down. What are the three major functions of the nucleus? The three major functions of the nucleus always. First is the major functions. We try to understand the prominent three major functions. Number one, it will help or regulate functions of the cell. Regulates the functions, all the overall functions of the cell. When I say functions of the cell, meaning all the other organelles, every organelle it can control. Number one, controls the process of cell division. Controls the process of cell division. When should the cell divide? Into how many daughter cells it should divide? Everything by the nucleus. Third, regulates the inheritance. Inheritance means just now that I told you about the transference. Inheritance of characteristics. Regulates inheritance of characteristics from parents to offsprings. From parents to offsprings. Offsprings means the next generation organism. So these are the common functions of the nucleus. What are the common functions of the nucleus? Nucleus will always perform all these functions. Next, after the nucleus, let us talk about one more cell organelle, and that is the endoplasmic reticulum. To talk about the endoplasmic reticulum, I am trying to show you certain images of the endoplasmic reticulum. How does it look like in the cell? This endoplasmic reticulum is absolutely in the form of some tubular structure. Let me just draw some of these. Endoplasmic reticulum generally would appear like this. Check if you have drawn in your diagram. If not, you have to draw. Check in your diagram if you have drawn. If not, complete drawing these structures. So there are one type of endoplasmic reticulum which will be connecting the nuclear membrane directly to the cell membrane, forming a pathway like this. And I am drawing only one. This type of structures in the cell are many. So if this type of tubules inside the cell are many, then is it not helping forming the shape of a cell framework? Yes. This is a tubular network, double-walled tubes. Can you see these are all double-walled tubes? A similar structure see there. So these type of tubular structures known as the endoplasmic reticulum. But prominently they are present in two forms. Now if I have to draw some other, in certain cells or in the same cell, sometimes both the types of endoplasmic reticulum is present. So sometimes this endoplasmic reticulum would be in this form, and sometimes they would be in some other form like this. So that means I'm saying that not necessarily every time they should remain connected with the end of the cell membrane and the nuclear membrane and the cell wall. Not necessarily every time they should be like this. Sometimes they must be just present in the cytoplasm as small, simple units. But whatever it is, the moment I talk about the endoplasmic reticulum, what are their features? The endoplasmic reticulum, what are their features to understand? What are their structural features? What are their functional features? Talking about the next organelle, that is endoplasmic reticulum. This endoplasmic reticulum that we are talking about, let us learn some features about the structure first. About the structure, I said the endoplasmic reticulum, what is the first feature to remember? The endoplasmic reticulum is double walled. It is a network of double walled tubular structures. So all over, it is a network of double walled tubular structures. Network of double walled. Always remember, fill in the blanks and all this also statements when you are answering. Network of double walled tubular structures. Now, this network of double-walled tubular structures, where are they present? They may be present, location can be two locations. One is connecting the nuclear membrane to the cell membrane, or only present in the cytoplasm. So, these tubular structures may connect the nuclear membrane and the cell membrane. They may connect the nuclear membrane and the cell membrane, or they may be only present in the cytoplasm So this is their location. Where are endoplasmic reticulum present? Next we talk about the types. What are the types of the endoplasmic reticulum, and based on what are we talking about the types? One type of endoplasmic reticulum, I may find that on the outer surface of this endoplasmic reticulum, there are some granular structures all over. One type of endoplasmic reticulum, I see there are granular structures all over. So what is this endoplasmic reticulum known as? This endoplasmic reticulum will be known as rough endoplasmic reticulum. This will be known as rough endoplasmic reticulum. What about the other? The other I see in the cytoplasm present, but they don't have those granular structures surrounding. These granular structures that I am talking about may be present separately also, individually also in the cytoplasm. So what are these granular structures otherwise known as? These granular structures otherwise are known as ribosomes, which itself is another cell organelle, but a very small minute cell organelle. So, what is the other type of endoplasmic reticulum which does not have these granular structures? This type of endoplasmic reticulum will be known as the smooth endoplasmic reticulum. So that means how many types of endoplasmic reticulum are there in the cells? Two types of endoplasmic reticulum. And what are their types and how are they different? So I talk about two types of endoplasmic reticulum. One is RER and the other is SER. What is the meaning of RER? RER means rough endoplasmic reticulum. SER means smooth endoplasmic reticulum. Structurally, what is their major difference? Both of them are tubular structures. Both of them are double-walled tubular structures. But difference कहाँ पे है? They possess ribosomes. They possess ribosomes on their outer surface. They possess ribosomes on their outer surface. That means endoplasmic reticulum which has ribosomes on the outer surface is known as rough. Does the SCR possess ribosomes? No. They do not possess ribosomes. They do not possess ribosomes. Are you following? They do not possess ribosomes. Following? So now that we are talking about what is the major criteria, how are we dividing the types of endoplasmic reticulum? Whether ribosomes present or not. Ribosomes present, then what are we calling them? Rough. Absent, smooth. But now that I talk about, it is possible that in certain cells, this rough endoplasmic reticulum, they are more in number. In certain cells, smooth endoplasmic reticulum, more in number. So we generally would not come across cells in our body where they are generally equal in number based on the function. वैसे तो सारे cells में smooth and rough would be there, but in certain cells rough is more and in certain cells smooth is more. तो कहाँ पे and what are their functions? Rough endoplasmic reticulum के functions हैं to synthesize proteins. That is their function. Make proteins, prepare proteins in the cell. That is their function. Synthesize meaning to produce. So what is the function of RER? To synthesize proteins. What is the function of SER? The function of SER would be to synthesize fatty substances. I would say synthesize lipids. The word lipids and proteins you are already aware with reference to the cell membrane. You have to write all these things that I am writing. लिखते रहो. लिखो, सुनो and clarify.
The function of ribosome.
The function of ribosome is to produce proteins.
No. In one different paragraph, not the RER, nor the SCR. That line in the middle.
The function of ribosome is to produce proteins. That's the reason whenever RER is present with that's the reason when endoplasmic reticulum is present with ribosome, they are producing proteins. Otherwise, endoplasmic reticulum, अगर मैं सिर्फ endoplasmic reticulum की बात करूँ, they are not responsible for making proteins. They have got some other functions. But यही endoplasmic reticulum जब ribosomes के साथ present होता है, its function is to make proteins. Otherwise, they will be making lipids और भी इनके बाकी functions है. Understand it? So separately, if I talk about what is the job of this organelle, its job is to make proteins. What type of cells will majorly have RER? I can give one example like the muscle cells. क्योंकि muscle cells में proteins की बहुत ज़रूरत होती है. Whenever we talk about the process of contraction, muscle cells have a high concentration of RER. Then what about the lipids? Liver cells have a high concentration of SER. So based on, I just now told you that in certain cells we will come across high numbers of RER, in certain cells high number of SER. But then ये तो हो गया specific RER and SER के functions. Then otherwise, what is the general function of the endoplasmic reticulum? What is the endoplasmic reticulum doing? Look here, in this figure, if I show you, can you see on the outer wall there are some granular structures attached? If you get to see a figure like that in your paper where some granular structures are attached, clearly to identify this is RER, rough endoplasmic reticulum. But not necessarily every time it will be like this. Sometimes you may also come across that the endoplasmic reticulum is like this. Tubular structures है, but on the outer wall there is nothing. Can you see the outer wall is smooth? On its outer wall there is no granular structure attached at all. अगर outer wall में granular structures नहीं है, then this is SER, smooth endoplasmic reticulum. But majorly when we talk about endoplasmic reticulum, they are rough endoplasmic reticulum is more common. What are the common functions? Let us learn now. What are some of the common functions of endoplasmic reticulum? General functions क्या है? Since they are a tubular network, they provide structural framework. They provide structural framework, meaning give shape to the cell. They provide structural framework to the cell, just like our bones. Structural framework to the cell. They provide structural framework to the cell. Next is they help in transport of cell secretions. Transport of cell secretions within the cell or from one cell to another. What is the third? They help in formation of the cell membranes and the plasma membranes. Cell membranes and plasma membranes in a new cell. Okay, so I'm talking about 5 functions, remember. Apart from these 2 functions, this is, I would say this is number 4 and this is number 5. So 3 general functions. What are the 3 general functions I'm talking about? I'm saying, since it is a tubular network, now just think, if this is a tubular network, yahan pe bhi present hai, yahan pe bhi present hai. I'm just drawing one, but it is not only one present in a cell. No, there are so many present in a cell. So since this tubular network, so many present in a cell, will this cell be able to collapse very fast? No, it is almost giving a shape to the cell, structural framework. At the same time, can you see it is helping in the formation of a passage? So, काफी सारा substances from outside the cell can enter and help in its transport and to enter into the cell, diffuse to the membrane and enter into the cell nucleus. There are so many substances from the cytoplasm will go out of the cell. Since it is in the form of a passage pathway, the endoplasmic reticulum also helps in transport of substances in and out of the cell. Also, it helps in transport of substances within the cell. After this, we are going to learn the Golgi. So, Golgi mein what I am going to explain you. Golgi is another type of a cell organelle like this. Golgi is another type of a cell organelle like this. So, this type of a cell organelle in the form of a plate, one after the other in the form of a plate. So, now if I understand that this smooth endoplasmic reticulum has prepared what is it supposed to prepare? Lipids. So, it will make lipids, and then these lipids will be carried in the form of small packets. क्या बोलेंगे packets को? Vesicles. So these vesicles will now move towards the Golgi. It will go towards the Golgi, fuse हो जाएगा Golgi के साथ, and then the secretion will enter into the Golgi for processing.
They would be carried by vesicles.
हाँ, जो भी secretions है, these are not like molecules floating in the cytoplasm. They will be carried in the form of small packets. जैसे by the flow of the cytoplasm, because this is a streaming substance, gel-like substance है cytoplasm. It is not stagnant. ये भी थोड़ा move हो रहा है constantly in the cell. So if I talk about the proteins, say suppose now this will make the protein molecule, which will be carried in the form of vesicles. Whether I talk about this or I talk about this, they will always produce proteins or lipids. But how will the protein lipid go from one part of the cell to the other? In the form of small packets. उस पैकेट को क्या बोलेंगे? Vesicles. So इस तरह से this vesicle may go and fuse also with the cell membrane and throughout the secretion outside the cell. ये भी possible है. So they may carry their secretion outside the cell, or they may carry their secretion from one part of the cell to the other to be used up in the cell membrane. समझो the cell membrane has gone through some injury, repair की ज़रूरत है. Then these proteins will go and build up the cell membrane. The nuclear membrane. The cell membrane is strong. Then what will happen? The proteins will come up here, the lipids will come up here, and it will build up the cell membrane. Are you understanding? So in the entire process of the repairing of the nuclear membrane and the cell membrane, maybe ये protein and the lipid will be useful. They will go out of the cell, they will keep on distributing themselves inside the cell in different forms. What is the third function? The third function is, this is what I just now mentioned, formation of the cell membrane and the plasma membrane. And apart from that, synthesis of proteins and lipids. So 5 functions of the endoplasmic reticulum we have come across. It helps in transport, it helps in providing structural framework, and it also helps in repairing the cell membrane, plasma membrane, formation of lipids, formation of proteins. Now, related to the plasma membrane, let us try understanding what is the Golgi doing. What is this Golgi doing? How about the Golgi in a cell? The Golgi complexes, the name Golgi complex. Whenever we are talking about the word Golgi, what is the first thing that should come to your mind is, we are talking about the Golgi complex, and the Golgi complex and the endoplasmic reticulum. How are they related? Let's try understanding. The endoplasmic reticulum and the Golgi. The endoplasmic reticulum and the Golgi, how are they related? The Golgi is always constantly processing. If I talk about this, the Golgi actually looks like this. Let us have a look at this Golgi. How does the Golgi look like? The Golgi looks like this. So Golgi looks like this. If I now have to draw simple, these are just small satellite structures in the form of stacks. See they? Some 10, 15 of them together always. So can you see those small bubble-like structures जिसके आसपास है? These are the packets containing the secretions. And either they go and fuse or they are formed. So ये constantly endoplasmic reticulum में भी होता है, and the Golgi also. See these are known as the vesicles. So this may be a vesicle which is coming from the endoplasmic reticulum and fusing, or this may be a vesicle developing from the Golgi itself. Clear? So now let us try understanding a relation of the endoplasmic reticulum and the Golgi. What is this Golgi doing? What is the next organelle we study? Golgi complexes. Golgi bodies or Golgi complexes. We talk about the Golgi bodies or the Golgi complexes. Golgi complexes, when I talk about, they are also double wall. It's a network of double wall, not tubular structures, but these are sac-like structures. These are not tubular structures, these are sac-like structures. See, disc-shaped structures there. Disc-shaped structures, and they are not connecting actually the cytoplasm, the nuclear membrane and the cell membrane, but they are just present in the cytoplasm. Yeah. Just present in the cytoplasm. Most commonly they are found between the nucleus and the cell membrane, but they don't form a pathway or else. They are just present this way. Okay. Now, are we talking about their types? No, we are not talking about any types. Golgi does not have any types. Golgi does not have any types as such. Endoplasmic reticulum had types, but Golgi does not have any types as such. But then, since we are talking about the structure, the Golgi has some extensions. When we are talking about the Golgi, if you see the sac-like structures that they have, what are those sac-like structures known as? Those individual sac-like structures can also be termed as what? The individual sac-like structures can also be termed as the cisternae. What are cisternae? The tubular sac-like or the individual sac-like structures are known as the cisternae. Cisternae of the Golgi. What is the meaning of cisternae of the Golgi? The sac-like structures of the Golgi. Now, talking about the function, what does the Golgi produce? I may say that this Golgi is producing so many different type of enzymes. The Golgi is supposed to produce this entire network of satellite structures that we understand. This entire network will be producing so many different type of enzymes. So many different type of enzymes. But here, we are also to understand that there will be some different types of enzymes produced by the Golgi. So, सबसे important function that Golgi goes through is secretion of enzymes. ये सबसे important function है of the Golgi. Secretion of proteins and enzymes. Secretion of proteins and enzymes. That is the most important function. Second. So I would say
all after secretion is good for the cell.
yes, the cell is inherently secreting those substances. So the secretion of the enzymes and the proteins, almost 40 different types of enzymes Golgi can produce. Almost about 40 different types of enzymes the Golgi produces. So almost about different types. Now whatever the major type of enzymes that the cell produces. Now see, quite a number of proteins are we are consuming along with the food. And there are variety of enzymes and proteins that the cell makes on its own. So the Golgi can produce a variety of such type of enzymes and proteins on its own. Next, what is its other function? Transport, I'm sorry. Packaging and transport. Packaging and transport of the cell secretions. Packaging and transport of the cell secretions. Packaging and transport of cell secretions, meaning, try to understand this. One point I told you, releasing, producing. One function I told you, that is producing what? Different type of enzymes and proteins. That is the primary function. Second function is what? Now that the endoplasmic reticulum is producing the lipid molecules, what will happen to this? Is this lipid directly released from the endoplasmic reticulum and showing its function inside the cell? No, it cannot. It has to go through the Golgi. So what will happen? This molecule containing the lipid, this vesicle containing the lipid will go and fuse with the Golgi. अब ये जो भी secretion है, this will go and fuse with the Golgi, and as it goes and fuses with the Golgi, what will happen? The secretion is all inside the Golgi now. After the processing, modification of the enzyme or the protein, whatever the Golgi has, whatever the endoplasmic reticulum has produced, the molecule will go through the Golgi, and अभी यहाँ पे क्या हो रहा है? Modification हो रहा है, processing हो रहा है. This will again give rise to one more bubble-like structure, and now this bubble-like structure is containing the lipids, which is capable of showing its function. This is now active. This is the active enzyme, meaning it is now capable of showing its function. So what was the job of the Golgi here? What did the Golgi do here? It did the process of, it did the function of packaging, processing, and then its transport. So what is the meaning of the word packaging? We always write packaging cell secretion, a prominent function of Golgi. Whenever the cells are producing certain chemical substances, it is always in the raw form. जो functional नहीं है. तो functional कब होगा? It has to go through a factory. It has to go through an industry जहाँ पे उसका processing होके final packaging होगा, and then now it will be released, and now this molecule can show its function inside the cell or outside the cell. So that is the reason we say the Golgi plays an important role in packaging. Packaging what? The cell secretions, the different type of cell secretions, whatever the cell had prepared. I talk about secretions of ARIA, secretions of SER, or anything else. Everything has to go through the Golgi, and then come out as active and processed form. Okay, that is cell secretion. It also helps in transport of these cell secretions. But now, when I say that the first function, what did I write? The first function is produces variety of enzymes. It is producing 40 different types of enzymes, but there are one specific types of enzyme which is containing some hydrolytic function, meaning which are very strong enzymes can dissolve the cell membranes. Golgi is producing 40 different enzymes, but उनमें से एक दो type of enzymes बहुत strong रहता है. Those enzymes are known as digestive or hydrolytic enzymes. तो वही सारे vesicles which will contain the hydrolytic enzymes उनको बोलेंगे lysosomes. So Golgi will produce so many packets like this. इस तरह के बहुत सारे packets Golgi is making. सारे packets में hydrolytic enzymes होगा? No. Only in some. So out of the 40 types of enzymes, maybe 3 types of enzymes are hydrolytic. So वो जो packets containing hydrolytic enzymes, those packets are known as lysosomes, which is a separate organelle altogether in a cell. So if this is the relation, then can I write Golgi gives rise to, or the lysosomes originates from Golgi? Yes, we are talking about the functions of the Golgi, so we will write gives rise to lysosomes, which are separate cell organelle altogether. Gives rise to lysosomes containing hydrolytic enzymes. Containing hydrolytic enzymes. So three major functions of Golgi क्या है? Three major functions of Golgi है. Golgi will help in secretion of different type of enzymes. Golgi will help in packaging of the cell secretions, उनका processing, उनका transport. And Golgi will produce special types of enzymes having hydrolytic function, digestive function. So those packets containing the digestive enzymes will be known as lysosomes. So I can write, yes, lysosomes are originating from the Golgi itself. Lysosomes are originating from the Golgi itself. अब ये जो lysosomes है, what are lysosomes carrying? Lysosomes are carrying digestive enzymes. Maybe now this cell is coming in contact with, or maybe some microbes are entering the cell. I talk about a lysosome. Say suppose I think of a lysosome separately. What is this lysosome containing? This lysosome is containing hydrolytic enzymes. The moment this lysosome comes in contact with the microbes, now say suppose these are the microbes. The lysosome is containing some hydrolytic enzymes, and these are the microbes. How will the lysosome behave now? The lysosome, just like an amoeba, the lysosome, what will it do? The lysosome will start engulfing all these substances, microbes, resulting in the formation of a structure something like this, where it will take in all these microbes. Ultimately, this lysosome will take it all in, and with the help of the digestive enzymes, now the digestive enzymes and the microbes come in contact, and the microbes and the digestive enzymes all will be destroyed. So this is almost behaving like an amoeba. Every single lysosome that we have in our body is constantly killing the disease-causing germ which is entering our cell every day. Every day we don't fall sick. Every day microbes are entering our body, our cells in hundreds. But how every day we are not getting infected, or we don't show any signs of infection? Because inside our cells, those hundreds of lysosomes are constantly eating away the germs. Then when do we fall prey to all these microbes? When do we start suffering from protection when the entry of the microbes is too much, or when the number of lysosomes in our cells are less, whatever the reason is. So at times only when the lysosomes are not able to tackle the microbes, then only we fall prey to infection. So then these lysosomes that we are talking about, then what are lysosomes? What is its function? What is the relation of the Golgi complexes and the lysosomes? Golgi complexes and lysosomes, they are absolutely related, but they are two different cell organelles, remember. But why they are always studied together? Because lysosomes are formed from the Golgi. That's why. Lysosomes are formed from the Golgi. That's why they are always studied together. Then, talking about the lysosomes, how do they appear? The lysosomes are very simple structures. They are very simple structures. Just one feature to remember, they are single-walled vesicular. They are single-walled vesicles containing what? They are single-walled vesicles containing digestive enzymes, which are also known as hydrolytic enzymes. And these lysosomes, they can become bigger by uniting with one another. So, this lysosomes, if I think of, yes, some small lysosomes, primary lysosomes can also fuse. Primary lysosomes fuse together to become secondary lysosomes. Secondary lysosomes. What is the meaning of primary and secondary here? Primary lysosomes here means that the small lysosomes, they sometimes come together and they become bigger inside the cell. The amount of the enzyme is so much and they can easily kill the microbes. So here the word primary and secondary is typically talking about how they tackle with the germs. So when we are talking about the lysosomes, remember the word that I mentioned? The lysosomes talking about the job of how the lysosomes can eat away the germs. Let's look into this figure. This is what I've been telling you. The lysosomes can become bigger. When they join together, they become bigger and result in the destruction of the germs. Here, about the lysosomes, one more concept you are supposed to study, why the lysosomes are known as suicidal bags. That's a very important question in your paper that you get. Lysosomes known as suicidal bags. We'll learn about the function in the next class.